Welcome to this lecture on developmental biology. Today, we're going to explore one of the most fascinating concepts in the field, fate maps. Imagine looking at a tiny, featureless sphere of cells and knowing exactly what each part will become: a beating heart, a complex brain, or the skin. This is not science fiction. It is the reality of embryonic development. By the end of this video, you will understand how scientists trace this incredible journey from a single cell to a fully formed living creature. To start our journey, imagine you are looking at a massive pile of building materials, like bricks, glass, wood, and steel. If you just stare at that pile, you have absolutely no idea what the final house will look like. However, if you have an architect's blueprint, it tells you exactly where every single brick and pane of glass will go. In developmental biology, an early embryo is exactly like that pile of materials. It consists of a tiny ball of seemingly identical cells. A fate map is the biological blueprint that tells us exactly what each of those early cells will eventually become in the fully grown animal. Now that we have our blueprint analogy, let us formally define what this means in biology. A fate map is a detailed diagram of an early-stage embryo that indicates the specific future tissues and organs that will develop from each specific region or individual cell. By creating these maps, scientists can look at a featureless sphere of cells and point to the exact spot that will become the brain, the heart, or the skin. It is literally a map of the future. Ultimately, a fate map provides a spatial and temporal lineage, connecting the simple geography of the early embryo to the highly complex anatomy of the adult organism. Understanding what a fate map is leads us to ask, how do we actually create one? To create these maps, researchers use various clever techniques to tag or label cells in the early embryo. One common method is injecting them with harmless fluorescent dyes, while another involves using genetic markers. Once the cells are tagged, scientists let the embryo grow and observe where those labeled cells end up in the mature organism. For example, if a researcher injects a dye into a specific cell on the top of a frog embryo and later finds that dye only in the frog's brain, they can map that original spot as the presumptive brain region. This process requires meticulous observation and tracking over time, often mapping the embryo in 3 dimensions. Having discussed how we tag cells, let us look at a classic visual example of a fate map. What you are seeing here is a diagram of a frog blastula, which is an early stage of the embryo. Notice how the sphere is divided into 3 distinct colored regions. The blue region at the top represents the ectoderm, which is destined to become the skin and the nervous system, including the brain. The red band in the middle is the mesoderm, which will eventually form the bone, muscle, and blood. Finally, the yellow region at the bottom is the endoderm, which will develop into the gut and internal organs. This diagram beautifully illustrates how specific regions of a seemingly simple sphere are already programmed to become highly specialized adult tissues. Now that we can visualize a fate map, we must ask, why do these maps matter so much in the real world? First and foremost, they are the absolute foundation for understanding how birth defects occur. If we know where a tissue is supposed to come from, we can trace back exactly when and where the developmental process went wrong. They are also crucial in the field of regenerative medicine and stem cell research. If scientists want to grow new liver or heart tissue in a lab, they need to understand the exact step-by-step pathway that early embryonic cells take. Furthermore, fate maps allow evolutionary biologists to compare how different animals develop, showing how evolution has tweaked the developmental blueprint over millions of years. Understanding the biological importance of fate maps naturally brings us to the analytical side of developmental biology. How do we mathematically represent the tracking of cells in a fate map? It's not enough to just say a cell becomes part of the brain. We need to know exactly how much it contributes. One common way is to calculate the lineage contribution percentage. This metric tells us exactly how much a specific early cell contributes to a final target tissue. By quantifying this, researchers can determine if a cell's fate is entirely restricted to a single organ or if its descendants spread out to form multiple different tissues across the body. To quantify this contribution, we use a straightforward mathematical relationship. The formula is C equals n sub d divided by n sub t, all multiplied by 100. Let us break down what each symbol means. The letter C represents the lineage contribution percentage, which is the final value we want to find. The variable n sub d represents the number of descendant cells found in the specific target tissue, like the brain or the heart. The variable n sub t represents the total number of descendant cells produced by that original early cell across the entire body. We're simply calculating a fraction and turning it into a percentage. If the value of n sub d increases while n sub t stays the same, the original cell is contributing more heavily to that specific tissue, increasing C. If n sub d equals n sub t, it means 100% of the cell's descendants went to that one tissue. Before we move on, let us see this formula in action with a concrete example. In the microscopic worm C. elegans, researchers are mapping the fate of an early cell called the AB blastomere. We're given that the AB cell has produced a total of 278 descendant cells. Out of these, 200 cells are found in the worm's nervous system. We need to find the lineage contribution percentage. We choose our formula and plug in the values. N sub t is 278 and n sub d is 200. Dividing 200 by 278 gives us approximately 0.719. Multiplying by 100 gives us our final answer of 71.9%. Physically, this means that nearly 3/4 of the AB cell's developmental energy is dedicated to building the nervous system, showing a strong bias toward neural tissue. Now that we have successfully calculated lineage contribution, we must address a practical limitation in the lab. When researchers inject a fluorescent dye into a single cell, that dye does not magically multiply. Every time the cell divides into two daughter cells, the dye volume is split in half. This means that as the embryo grows and cells divide repeatedly, the fluorescence intensity decreases rapidly. This physical reality creates a strict limit on how far into the future researchers can draw the fate map. If the dye becomes too faint for the microscope to detect, the tracking process halts. Therefore, we need a way to calculate how this dye dilutes over time to ensure our tracking methods are actually viable. To predict if our dye will remain visible, we use the dye dilution formula. The formula is I sub n equals I sub 0 divided by 2 raised to the power of n. Let us define these terms. The symbol I sub n represents the fluorescence intensity of the tracking dye after a certain number of cell divisions. The symbol I sub 0 represents the initial fluorescence intensity when the dye was first injected into the early embryonic cell. Finally, the symbol n represents the number of cell divisions, or generations, that have occurred. The number 2 is used as the base in the denominator because one cell divides into two daughter cells. As the variable n increases, the denominator grows exponentially, which causes the final intensity to decrease very rapidly. Let us apply this dilution formula to a real laboratory scenario. A researcher injects a fluorescent tracer dye into a single cell of a frog blastula. The initial fluorescence intensity is measured at 800 units. The microscope requires a minimum of 20 units to detect the dye. We need to find out if the dye will still be visible after 5 cell divisions. We use our dilution formula, plugging in 800 for I sub 0 and 5 for n. First, we calculate the denominator. 2 raised to the power of 5 equals 32. This means the original dye is now spread across 32 individual cells. We then divide the initial 800 units by 32, which gives us a final intensity of 25 units. Because 25 is greater than the microscope's threshold of 20, the researcher will indeed be able to see the cells and successfully draw the fate map. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that a fate map is an essential biological blueprint that reveals the future destiny of every cell in an early embryo. Second, we saw how scientists use clever tracking techniques, like fluorescent dyes, to draw direct lines from a featureless sphere of cells to complex adult organs. Third, we explored how to mathematically track these lineages using contribution percentages and how to account for dye dilution over multiple cell divisions. Mastering the concept of fate maps is your first major step toward understanding the beautiful, highly orchestrated symphony of embryonic development. Thank you for watching.